Welcome to Mindray Park. Mindray, as a global leader in the field of hematology, has been actively engaged in continual technological innovation. In 2021, we introduced our first self-developed automated digital morphology analyzer, NC80. In 2022, we launched our first CBC and ESR integrated analyzer, BC700 series. Thanks to these innovative breakthroughs, we have entered a lot of top-tier hospitals, gained recognition from customers worldwide, and helped them improve testing quality and diagnostic efficiency. Today, we are bringing innovation to the abnormal cell screening. While hematology technology seems to be very mature in the eyes of many people, in the era of rapid viral mutations, especially with the prevalence of respiratory diseases, there's a pressing need globally for a more efficient and accurate screening diagnostic tool to elevate the burden on healthcare systems. Furthermore, with the trend of malignancies affecting younger populations in recent years, as the industry leader, it is essential to prevent misdiagnosis during the diagnostic process, achieve precise preliminary screening, and ensure that patients receive treatment at the earliest possible stage. Next, let's witness together the unveiling of Mindray's next generation hematology analysis system.
Hi everyone, it's nice seeing you all here again. As an innovative leader in the field of medical devices, we provide more than just products. We focus on delivering intelligent, comprehensive solutions that cater to hospitals, laboratory technologists, pathologists, and patients. In reality, we face many challenges. Although there are differences between countries and regions, the overall trend shows an increasing number of tests in laboratories with higher demands for testing accuracy and abnormal cell screening. At the same time, issues such as limited space and staff shortages are becoming more common. To address these challenges, Mindray's latest BC7600 series automated hematology analyzers, along with CAL 9000, CAL 7000 cellular analysis line, offer various configurations to meet the diverse needs of our customers, aiming to deliver more clinical value with minimal space. Next, our colleague, Echo, will give a detailed introduction of this new generation of hematology analysis.
Thank you, Shan. Hi everyone, I'm Echo. This is our new hematology cellular analysis line, CAL 9000. With just a single EDTA tube, you can conduct multiple tests in a short turnaround time, supporting rapid preliminary screening. The new BC7600 series automated hematology analyzer alone can perform CBC, DIF, and ESR inflammation tests within 40 seconds. Unlike most existing setups in lab that require separate instruments, this integration saves space and provides more accurate results. This analyzer adopts the SS-Cube cell analysis technology. During preliminary screening, it utilizes data from the optical DIF channel and WNB channel to provide various parameters and 3D scattergrams. For the first time, we have introduced a disease-specific alarm for reactive lymphocyte due to infectious mononucleosis. This helps the laboratory screen for suspected acute mononucleosis samples, reducing misdiagnosis, effectively identifying infection types, and assisting in accurate clinical treatment. We have also introduced the alarm for abnormal promyelocyte. It can help in the early detection of acute promyelocytic leukemia patients, allowing them to receive treatment sooner and improving the cure rate. Here, I want to emphasize the brand new hematology Malignant channel. It provides a new three-dimensional scatter plot that offers a more specific detection for abnormal cells. At the same time, it enhances the specificity of blast cell and abnormal for myelocyte, significantly reducing false positive rates, decreasing the workload for further re-examination like microscopic review. For abnormal samples, slide preparation and staining can be performed and connected to the MC80 automated digital cell morphology analyzer. The new H120 automated glycohemoglobin analyzer can automatically switch between variant detection mode and fast mode, which is up to 120 tests per hour, providing accurate A1C results. When a single sample is sent for testing, our H120 analyzer can perform immediate testing without waiting, while reducing reagent waste. Reducing manual intervention does not compromise our meticulous attention to every sample at any stage. Through the TM1000 sample processing system, we have achieved a fully automated process from loading to archiving. A large batch of samples can be directly poured into the bulk input unit for automatic sorting and registration. The system intelligently matches the most suitable preliminary and retesting process based on the sample information from laboratory information system. After testing is completed, samples are archived in the corresponding area according to their sample types or features that could be user-defined for convenient access. For a small number of abnormal samples that require further action, you can locate the axis in the output area with just a few clicks. Our all-in-one solution offers accurate and comprehensive test results with a single EDTA tube, reducing the amount of blood drawn from patients and the waiting time for reports, allowing doctors to provide treatment plans earlier. Up next will be our colleague, Dr. Terence.
Hello everyone, I'm Terence. With over a decade of research and experience in hematology analysis, our new generation hematology analysis system can reliably screen abnormal samples and intelligently handle re-examinations with Mindray middleware labs expert. It also provides additional clinical information to assist in critical decisions, such as screening for blood-related disease and transfusion needs. Accurate PLT results are crucial for clinical transfusion decisions, especially for patients with low PLT counts, where the risk of bleeding is very high. Mindray has innovated in precise PLT detection, combining re-examination rules to provide a complete PLT solution. Our PLT Edge technology uses advanced algorithms to integrate information from both the impedance channel and the optical dip channel, eliminating interferences that traditional impedance methods struggle with, and improving accuracy without extra costs. When abnormal PLT samples trigger re-examination, the PLT-O technology under the RET channel ensures high precision in low-range detection and eliminates false low values with its platelet clumps deaggregation function. The system also automatically prepares slides and performs microscopic examinations. The PLT-N mode in our MC80 estimates PLT counts to confirm results, while the PLT Pro mode scans a blood smear edges and tail within 1 minute to rule out PLT clump aggregation. Through these innovative technologies and intelligent re-examinations rules, we have minimized manual intervention for accurate PLT results. Next, I would like to invite our senior product specialist, Kyle, to give you some idea how Mindray safeguards your results with our quality control system.
Hello everyone, this is Kyle. In this section, I am going to introduce you Mindray's fully automatic quality control system. The reliability of results depends on comprehensive quality control system. As laboratories take on more projects and deploy more equipment, the resources, time, and effort invested in quality control also increased. To address this, we have upgraded the TM1000 with a fully automatic quality control module. This module includes a refrigeration unit that stores the quality control material and can automatically start the cellular analysis line each day to perform quality control on all hematology analyzers. Traditional quality control platforms risk discontinuous monitoring. Even with multiple quality control checks each day, certain system failures can only be detected at the next scheduled check, making real-time monitoring impossible and challenging to identify affected samples. This requires retesting all samples from affected periods. To solve this, we introduced NNPBRT-QC. This technology monitors the test result based on sample information like department, gender, age, and disease. It can issue a failure alarm quickly and visually locate the affected sample, significantly reducing the impact of failures and subsequent corrective action. The fully automatic quality control module and NNPBRT-QC are just some highlights of our seamless and worry-free automatic quality control system. You will experience more benefit from our CAL 9000 in your actual work.
Hello everyone! It's truly an honor to welcome you to Mindray's Guangming R&D Center in Shenzhen. This is a place where we bring our mission to life, advanced medical technologies to make healthcare more accessible. As we know, accurate test results are the foundation of laboratory medicine. However, what really impacts patient diagnosis and treatment decisions is always the skills and experience of lab specialists. Today, as we stand on the wave of AI with the massive testing and patient data in clinical laboratories, questions arise. How can we utilize AI to amplify and extend the wisdom of laboratory experts? We deeply understand that the true medical AI should never be a substitute for human, but rather an indispensable component of human AI diagnostic centers. This drives our team to continuously explore and break through in hematology using AI technology. And today, you will see how we bring all of this to life.
Disease screening has always been at the heart of early disease detection, and hematology analyzers are essential in this process. But accurately interpreting the results, including parameters, flags, and scatter plots, can be tricky. It requires extra attention and expertise to guide further testing and eventually lead to a correct diagnosis. This process is not straightforward, especially when dealing with atypical patterns that require additional time and expertise. With the advancement of our hematology analyzer, the game is changing. We now have access to an enormous amount of data from hematology analyzers. And with the artificial intelligence expertise we've built over the past decade, we started asking, why not push the boundaries and use the hematology analyzer to flag diseases directly? Think about the possibilities it could open. Faster disease diagnosis, fewer uncertainty, and earlier diagnosis and treatment, especially for conditions require immediate intervention.
Our algorithm team has been studying various blood diseases using scatter plots. Among all diseases, the severity and rapid progression of APL truly impressed me. However, if APL cases can be detected early during peripheral blood screening, ultra treatment can begin even before a definitive diagnosis is made. This fast detection allows for early disease control and can increase the cure rate by up to 95%. That's why we first chose APL as the focus of our study. Our goal is to help clinical laboratories flag APL cases in the early stages, ensuring patients receive the treatment they need as soon as possible.
One of the biggest challenges we faced is that APL is a rare disease, making it extremely difficult to collect enough samples for research and development. On top of that, APL has numerous subtypes and stages, each with distinct characteristics. So, to build a robust database, we need to collect samples from all subtypes and stages of the disease. To overcome this challenge, we started collaborating with hospitals over 10 years ago to collect samples. And to date, we gathered 20,000 samples covering a wide range of blood disorders and hematology malignancies, including over 300 APL samples. This extensive sample collection has built a very solid foundation for the development of our APL flag.
With our abundant data, we first tried to build an APL screening model using both CBC parameters and commonly used cell population data. However, during development, we hit a challenge. Some samples of hematologic malignancies and severe infections were being misclassified as APL. Upon further analysis, we realized that these samples share similar characteristics with APL, making them hard to differentiate. With our extensive AI experience in digital morphology, we decided to integrate convolutional neural network and class activation mapping technologies to capture the subtle feature differences that were hard to distinguish for humans. Through more than 100 trillion computations, our model analyzed scatter plots from both APL samples and over 120,000 interfering blood samples. Finally, our model located 4 key regions and extracted over 20 important features from the scatter plots in both DIF and WMB channels. And for APL samples, it identifies some features that align with clinical practice. For example, in the DIF channel, the model found a unique pattern at the boundary between immature granulocytes and monocytes, where APL samples had scatters in the low size scatter and high fluorescence area. But what really set our model apart was its ability to uncover hidden patterns that were not previously recognized in clinical practice. For instance, in the WMB channel, the model observed scatter points in the high size scatter and high fluorescence area for APL samples. We then optimized our model by combining both CBC parameters and AI extracted features. Finally, with a significantly improved performance in sensitivity and specificity, our model met the clinical expectations for APL disease screening.
Multiple evaluation studies have been done and confirmed the high performance of the APL flag. In addition, we have further validated the real-life effectiveness of the APL flag by integrating it into the routine practice at the top-tiered hospitals. The data and feedbacks we've received are very positive. Here's one case really stands out for us. A young father came in the hospital with persistent gum bleeding. A CBC only showed low WBC and SGB, but no blast. So the results was not alarming. However, an APL flag was triggered, which alerted the lab team to take a closer look. So they initially analyzed one blood smear, but found nothing unusual. But just to be absolutely sure, they carefully examined 3 more blood smears, and on the last one, they found 2 abnormal promyelocytes. This result was communicated to the clinician immediately, and the patient was held for further testing, which eventually confirmed an APL diagnosis. So as you can see from this case, the APL flag is more than just a tool. It is making a real difference in saving lives and families. Looking ahead, our R&D team is already exploring how to use AI technologies to screen other diseases, including sepsis, TTP, malaria, and thalassemia. Our goal is to continue developing disease screening flags to save even more lives in the future.
For clinical laboratories, the morphology confirmation is absolutely critical for disease screening. The precise recognition of abnormal cells remains the most critical and challenging breakthrough in digital morphology analysis. Currently, leveraging AI technologies to enhance the accuracy of image recognition stands as the optimal solution. However, there are still many challenges to overcome before it can fully meet clinical expectations. Over a decade ago, our team embarked on the exploration of high-speed microscopic imaging and abnormal cell recognition. In 2014, we introduced fully automated slide preparation and staining equipment. It streamlines the workflows of blood smear preparation. 5 years ago, with innovative multi-depth fusion technology, our system standardized ultra-high-speed digital imaging in microscopic environments. This breakthrough not only captured images faster, but also accurately restored the pathological features of abnormal cells. Within 3 years, the devices equipped with this system exceeded 1,000 units globally. This is really a testament to the trust and impact of our innovation.
Over the past decade, our team has conducted several multi-center studies in China dedicated to collecting samples, even some rare blood and infection diseases, accumulating a million-level cell bank. We have published some research findings. Specifically, in studies on the performance of blood cell classification, we have achieved precise identification of 15 distinct types of WBCs, nucleated red blood cells, and platelet aggregates. Notably, the recognition accuracy for the blood cells exceeds 19%.
Many countries globally prioritize addressing anemia and parasitic diseases. We have conducted many regional real-world studies of abnormal red blood cell identification. Our work now enables precise recognition of all erythrocyte morphologies outlined by ICSH. We can systematically conduct qualitative assessments of red blood cell populations and have also discovered clinically significant cellular inclusions. Among these efforts, our research on malaria parasite identification stands out. Microscopic detection of malaria parasites remains the WHO-endorsed gold standard for malaria diagnosis. While malaria is endemic in Africa, Southeast Asia, and South America, China is virtually malaria-free. To secure sufficient samples, our R&D and the clinical teams have dedicated substantial time and resources to field operations in those regions.
Accurate red blood cells assessment requires identifying thousands of cells, yet detecting even one single malaria parasite defines a positive sample. To ensure no malaria parasite is missed in high-density RBC scenarios, we implemented an object detection network with large-scale distribution and attention mechanism. However, another challenge emerged. The features of malarial parasites are extremely subtle and easily misjudged, especially when RBC are adhered to PLT. This led to an increase in the false positive rate. To solve this challenge, we implemented the fine-grained classification network, which effectively distinguished malarial parasites from other interference, lowering the false positive rate. This feature is extremely beneficial for malaria screening. At one of our partner hospitals in Thailand, limited by the number of professional microscopists, around 10 malaria cases were detected annually before. After integrating our device into the existing morphological analysis workflow. With no change in personnel, the hospital could examine twice as many samples daily, detecting 2 to 3 cases per month.
Collaborating with experts and hospitals across China, we standardize the application of AI from different aspects, forming an expert consensus. This has enabled AI to truly integrate into practical workflows, and we are actively collaborating with international hospitals to promote global consensus.
The application of AI technologies in disease early warning and abnormal cell detections marks the start of bringing lab testing closer to clinic diagnostics. For the laboratories managing massive clinic data, AI technologies not only free up manpower but also amplify the expertise of lab specialists. Aiming to create an unmanned hematology analysis line, we have made great progress using laboratory analytics data. First, by intelligently adjusting test sequences according to different sample types and testing needs, we achieve optimal scheduling efficiency and further reduce the TAT. Second, utilizing AI to customize rules for auto-review and auto-verification has significantly improved the auto-verification pass rate, further reducing the burden of manual review. Last but not least, AI simplifies the deployment process and enhances the performance of VPRD-QC, integrating it into routine laboratory quality management and ensuring consistent results among multiple devices within and across the laboratory. Looking ahead, we will continue to explore the interpretation of multimodal testing data and patient information. By applying large language models to internalize clinic thinking, we aim to enable intelligent report interpretation and diagnostic recommendations. This will push the evolution of laboratories from data factories to human and AI diagnostic centers, creating Mindray AI-empowered diagnostic workflows for better health care.